At the beginning, tyrosine kinases are individual polypeptides with ligand receptors outside the cell and tails with tyrosines on the inside of the cell. When the signaling molecule binds to the receptors, the polypeptides come together to form a dimer. This process, called dimerization, activates the tyrosine kinases in the tails of the polypeptides. These tyrosine kinases add a phosphate from ATP to tyrosine on the tail of the polypeptide. The ATP is now an ADP. The tyrosine kinase receptor is now fully activated. This is recognized by specific relay proteins. Before a relay protein binds to a phosphorylated tyrosine, it is inactive. When it binds to the phosphorylated tyrosine, it has a structural change and is activated, triggering the transduction pathway. In this instance, the cellular response is to synthesize protein. The G protein, an integral protein attached to the cytoplasmic side of the membrane, acts like a switch, turning on or off depending on whether GDP or GTP is attached to it. When a signaling molecule, or ligand, binds to the G protein coupled receptor, the receptor changes shape and GTP displaces the GDP, activating the G protein. After the G protein is activated, it dissociates from the receptor. The protein will then move along the membrane and attach to an enzyme. Once the protein is bound to the enzyme is activated. This will allow the enzyme to start the next step in the transduction pathway. The enzyme, now activated, catalyzes the conversion of ATP to CAMP. The CAMP now becomes a second messenger, activating a new protein kinase. The protein kinase then goes on to activate the signal transduction pathway. In this instance, the cellular response is the release of calcium ions into the cytoplasm. In a healthy cell, glucose enters the bloodstream as food is digested. The pancreas recognizes this and releases insulin into the blood. Insulin acts as a ligand, opening a protein channel that allows glucose to enter the cell. Glucose can then be converted for storage or used to make ATP. In an unhealthy cell, glucose enters the bloodstream after digestion, just like a healthy cell. However, the pancreas does not release insulin. Glucose cannot enter the cell, and the cell cannot function without ATP. According to the Mayo Clinic, common symptoms of type 1 diabetes are increased thirst, frequent urination, extreme hunger, weight loss, and blurred vision. To treat type 1 diabetes, synthetic insulin is injected into the bloodstream. The cell will recognize the synthetic insulin and will allow the glucose to enter the cell to create ATP.